Hello friends, welcome to my YouTube channel Advanced Bio Tutorial. Today I am going to discuss a topic that is cytokines. It is a topic of immunology and this topic is very much important for the students of graduation level, PG level and in different types of competitive exams and different types of entrance tests like NEET or university level admission test, etc. Okay, let's come to the topic. Cytokines, right? Come on to the etymology. If we break down this word cytokines, then we can find the cytos. That means cells, and kines. This word kines comes from the word kainos, which means the movement. That means cytokines means the substance which is moved from any cell. That is, it is nothing but the cellular secretion. Okay. Now come to the definition of cytokines. Before this, you should understand the meaning of keywords. First one is the biochemical substance. What does it mean? Biochemical substance. That means the chemical substance. Cytokines is a chemical substance which is synthesized within the living cell. That's why biochemicals. Okay. Now, second point is source and target. Source means, suppose cytokines is secreted from one cell. This is the C1 cell. Okay. Cytokines is secreted from here. And target is also another cell. That is the C2 cell. That is also the cell. This secretory substance will act on these cell. So, this is the source and this is the target. Okay. So, both are the cells. So, this substance helps in the communication. That is, it is nothing but the cellular communication. Okay. Every communication has a topic. This communication also have some signals or some message or some instruction. Why for this instruction? Come. This cell secrete this, that is cytokines, which act on this cell, that is the target cell. These substance carrying some information, and to receive that information, this target organ will act accordingly. So the message for the target cell is nothing but the what to do. Okay, now what this target cell will do after receiving these cytokines? These will act in different ways. Suppose proliferation. After receiving this will divide like this. That means multiplication. So this is one mode of work. And another is, suppose after receiving these cytokines, it will secrete another cytokines for the another cell, C3, another cell, to do something else. So this is also another mode of work. Now from this keyword, now we construct the definition of cytokines. Come on. Cytokines are the biochemical substances which are secreted from the cells of our immune system and carry some informations or messages or instructions for locally distributed cells. Those ultimately do accordingly as per the instruction. Okay, now it is very much simple to construct the definition from these keywords. Now, we can say that this cytokines is nothing but a chemical messenger like hormones, and it carries some signals also. So, we can say cytokines are the stimulating molecules or stimulating substances. Okay, now come to the another point, that is the properties of cytokines. First point, chemical nature. We know these are the biochemical substance, okay? But these are basically protein or peptides or glycoproteins by nature. So we can say these are the macrobiomolecule. Macro means the molecular weight of any cytokines is heavy, so it cannot move distantly. Now come to the point. It acts locally. That means over a short distance. Okay. Now, third point, that is the time span of their activity. It is very short. Why? Because if the time span of their activity is long, then what happens? The target cell will do any activity in repeated way. So there may create some problems. That's why time span is very short. Now, the working concentration. This can affect on another cell at a very low concentration, like the hormones. And next point is receptors of the cytokines. Suppose this is the secretory cell. So this is the cell 1, cell 1. Okay. And from here, two types of substances are released. Suppose this one. These are the cytokines. And this is the target cell. Cell 2. That is target cell, and this is the source. On the surface of this target cell, you can see the specific receptor of the specific cytokines. Right? Suppose another cytokines is released from here, and the structure is like this. Okay, like the square. It is to make you understand. In this case, the surface molecule or the receptors of these cytokines will be like this. That means the structure of one receptor is like this, and the structure of another receptor is like this. This is for this receptor is for this, and this receptor is for this. So there is the specificity of the receptors. Okay, that's why we can say for this point, the target cell of any cytokines have specific surface receptors for the specific cytokines. Now, last point, what type of signal it carries? That means cytokine carries the signals or informations that includes proliferation of the target cell. That informations may be for the secretion of another type of substances. Okay. So these are the signals. It may be to recruit another cells at the site of infection. Okay. There are so many diversities of work. Now come to the another point of the cytokines. That is the nomenclature of cytokines. See, cytokines is a generalized term. That means cytokines is secreted from one cell and act on another cell. Right? But it has some synonymous words. That means the alternative namings of cytokines are given on the basis of two things. See, on the basis of the name of the target cell. On the basis of the name of the target cell. 1. Number 2, on the basis of their mode of activities. How it works. Now come to different nomenclature. Lymphokines. First name. That means this is the source, a cell, and this is the target cell. The name of the target cell is lymphocyte. So when the cytokines is secreted from any cells and act on any lymphocyte, then that particular cytokines is known as the lymphokine. Okay. Now second one, monokines. When one cytokines secreted from any cell and act on monocyte, These cytokines is known as the monokine. Now, third, chemokines. Mostly all are the chemokines. It is based upon the mode of activities. That means the cytokines which shows the chemotactic activity. Okay. Now, fourth one, interleukin. Here is the S cell and here is the target cell, T cell. Now, the S cell is the leukocyte and the target cell is also the leukocyte. So, we can tell here the cytokines which is released from any leukocyte and act on another leukocyte is known as the interleukin. Clear? Now, fifth, autocrine action. These all are the based upon the modes of the activities. Suppose we have to mark here. These are the cytokines like this. Okay, these are the cytokines. Okay, and now see one thing. This is the autocrine activities. This is the source cell, and from here any cytokines is released, and it will act on what? On this cell. That means here the source cell and the target cell are the same. So these kind of work is known as the autocrine action. Okay. Now paracrine action. Suppose this is the source cell and it releases some cytokines and it will act on locally placed cells. That means adjacent cells. Okay. Now endocrine. This is the source cell. See here what is the distance. This is the local. The distance is short. This is the source cell and these are the target cell. The cytokines released here from here. See the distance. So here the distance is long. This type of work is known as the endocrine action. Okay. These all are the different nomenclature of the cytokines. I think it is clear to you. Okay. Now I am going to discuss the another aspect of the cytokines, that is the mode of actions of cytokines. You know there are so many varieties of cytokines and this variation also evident in their activities also. Now see, this is the mode of action. Basically 5 types of mode of actions. Okay. Number 1 is the pleiotrophy. Number 2 is the redundance. Number 3 is the cascade. Number 4 is the synergistic, and number 5 is the antagonistic activity. Now come to the first one, that is the pleiotrophy. Okay, now see, cell 1 is the secretory cell. From here the cytokines will be released, and here the C2, C3, C4, these are the different types of cells. Okay, those are the target cell, and which I am pointing now, that is the cytokine, red triangle. See, this cytokine will be released from the C1 and then these cytokines will be bound to the surface receptors of cell 2, cell 3 and cell 4. Now see, after receiving the signals from the cytokine, the cell 4 undergoes the proliferation and cell 3 involves in the differentiation. So we saw that their actions is different. Now in case of cell 2, after receiving that, the cell 2 secrete the another type of cytokines. Okay. Now here what happens? Same cytokine acts on different types of cell, resulting in different actions. So we can conclude that same cytokine, different target cell, different actions or effects. Okay. Now this is the example I am citing here. Now see this. Activated T cells secrete the interleukin 4, and these interleukin 4 act on 3 different target cells. Those are B cell, thymocyte and the mast cell. Okay. And see the activities. Mast cell involved in proliferation, and the thymocyte also proliferate, and the B cell will be activated and then proliferate and then differentiated. Okay, these are the different actions or effects are seen by the effect of same cytokine on different target cells. Okay, now this is the second one, redundancy. Now see here three types of cytokines. Cytokine 1, blue ball, cytokine 2, red triangle, and cytokine 3, yellow mass. Okay, now see the target cell is same, that is cell 1. Now cytokine 1 binds to the cell 1 and it will differentiate to another cell. It is the differentiation. Okay, it is the differentiation. And now we'll see the cytokine 2. Cytokine 2 also binds to the cell 1, and after receiving the signals it will undergo the same activity, that is the differentiation. And in case of cytokine 3 on same cell, that is cell 1, the action is same. So we can conclude from this animation that different cytokines act on the same target cell and the activity is also the same. Right? Now this is the example. See this. This is the activated TA cell secrete different types of cytokines, interleukin 2, interleukin 4 and interleukin 5. Okay, and it will act on B cell, resulting the proliferation. Activity is same, target cell is same, but different cytokines act on it. Multiple cause but same effect. It is just opposite to the prior one, that is the pleiotropism. Okay, now see the third one. This is the cascade activity, and this is the example I am coming here later. See here three types of cells are seen. Cell 1, cell 2 and cell 3. From cell 1 green cytokines are secreted, and which influences the cell 2 to secrete the another cytokine, and those cytokine is for the cell 3. Okay, now we can say in another word, the cytokine secreted from the cell 1 stimulates the cell 2 to secrete the another cytokine. So this is the cascade activity. Now see, this is the fourth one, that is the synergistic activity. Here it is written the two or more cytokines acting together. See, within the cell 1, though this is the secretory cell, three types of cytokines are there. This one, blue ball, okay, the red triangle, also the second type of cytokines, and the green mass, green rhombus is the another type of cytokine. There are basically three types of cytokines act on the cell 2, that is the target cell, and see what will be the effect for this. Right, see, the effect is the proliferation. Okay, and see the rate of proliferation is much more higher than the normal rate. What is the cause of this? This is due to the exaggerated effect. That means the combined effect. Because here the three types of cytokines simultaneously act on the target cell. For that the effect will be multiplied, and that's why the proliferation rate will be higher. Now come to the last one, that is the antagonism. Here you can see two types of cytokines, that is cytokine 1 and cytokine 2. Red is the cytokine 1, blue is the cytokine 2. And see, these cytokines, these two cytokines are secreted from the cell 1, and here the target cell is cell 2. Okay, Now let's see the animation. Now cytokine 1 is secreted and some molecules of cytokines bind to the target cell, but some cannot bind to it. Why? Because the cytokine 2 opposes it. The molecules of cytokine 2 blocks the molecules of cytokine 1 by masking it. Okay, this is the inhibitory effect. Now see this animation once more. See. Now see, this cytokine 2 masks, this cytokine 2 masks the cytokine 1. For that, the cytokine 1 cannot bind to cytokine 2. Ultimately, the effect will be reduced. This is called the inhibitory effect. Here, the one cytokine opposes the another one. That's why this type of activity is known as the antagonistic activity. Now, see the example. See, the activator T cell secrete the interleukin 4 and it also secretes the interferon gamma. Interleukin 4 cause the differentiation of B cell, but interferon opposes the activity or inhibits the activity of interleukin by blocking this. So the rate of differentiation will be reduced. This is the inhibitory effect or antagonistic effect. Okay, now I am coming to the another aspect. This is the most important one, that is the cytokine receptors. And before starting this, I want to say something about the fundamental concept of the cytokines. See, this is the cytokine receptor, and this is basically the transmembranic receptor, you can see, and here, this membrane, the pink color, this membrane is the membrane of target cell, and this portion of the receptor is the interior domain, and this portion of the receptor is the extracellular domain, extracellular portion. And here you can see this is the dimeric form or receptor, and you see there the two domains, top domain and the bottom domain. If you consider the bottom domain, then you can find the polypeptide chain like this, and here you can see the red line. What is this? This is the red line where the conserved sequence of amino acids is present. This feature determines the specificity of the receptors. Okay, now here on the top and the bottom domain you can see one such conserved sequence, and in case of second figure you can see in each domain there are two conserved sequences. Okay, now see the bottom screen. These are the two cytokine receptors on the membrane of the target cell, and just prior to the binding of the cytokines, those become closer to each other like this. See this. It seems to be that this is one receptor, but not that. This is one receptor and this is the another receptor. There are basically two receptors altogether here. Now, by taking this concept, you see this slide. There are basically 5 types of families of cytokine receptors, you see. Okay, and you can see the type 1 cytokine receptors, type 2 cytokine receptors, TNF. TNF means the tumor necrosis factor. TNF receptors family, and fourth one is the IG, that is immunoglobulin type of receptor family, and last one the G protein coupled receptor family. Okay, now the another name of the type 1 is the hemopoietic receptor, or it is also known as the interleukin receptor. See the receptors, the structure of this receptor is more or less like the before. Okay, see here two receptors are there. Okay, and here you can see the yellow line. These are the conserved sequences. Okay, at the top domain there are so many conserved sequences, but see, there is no conserved sequence in the bottom domain of the exterior site. Okay, now now if you see the examples of it, then you can see interleukin 2 to interleukin 7, interleukin 9, 11, 12, 13, 15, and colony stimulating factors. So many cytokines are there, those can bind to this type of receptors. Okay, now come to the second one, type 2 cytokine receptor. Okay, the structure of this receptor is more or less similar to the type 1, and the alternative name of this receptor is the interferon cytokine receptor family. Okay, now you can see both the top domain and the bottom domain of this receptor possesses the conserved sequences, where the type 1 lacks the conserved sequence in the bottom domain. Okay, this is the basic difference between two. Okay, now come to the third one, that is TNF receptor, that means tumor necrosis factor receptor family. And see, this is much more bulky than the others. Okay, if you see the exterior side of this receptor, then you can find those are tetrameric form. That means 4 domains are there at the exterior side, and in each domain there are so many conserved sequences. Okay, and if you see the examples, TNF alpha, beta, some growth factors, FAS, etc., etc., these cytokines can bind to this type of receptors. Okay, here 3 receptors are shown all together. Okay, now see the fourth one, that is the immunoglobulin type of receptor family, and this receptor is somewhat different from the others because this is made up of single polypeptide chain, and it also forms different domains with the help of the disulfide bond. I am writing here, this is the disulfide bond, 2 dots representing this. Okay, if you see the examples, then you can see only 3 cytokines can bind to this. Interleukin 1, monokine colony stimulating factors, and c-KIT. So we can tell that this receptor family is very rare. Okay, this is not very common. Now come to the last one. This is the G protein coupled receptor family. But the popular name of this receptor is the chemokine family of cytokine receptors. Okay, now if you see the structure of this receptor, then this is quite different from the others. This is the transmembranic receptor with multiple helix. Okay, now see, I'm denoting the transmembranic sites 1, 2, 3, 4. This is the 5, 6, and last one is the 7. So we can say the 7 transmembranic domain present in this type of receptors. Okay, if you see the interior portion of the receptor, the G protein is present here. Actually, G protein helps in the signal transduction of the cytokines. Okay, now see the examples of these chemokines can bind to this type of receptors. These all are the 5 families of cytokine receptors. In this video, we come to know that what is cytokine, the properties of cytokine, the nomenclature of different cytokines, and the mode of actions of cytokines, and the different types of receptor molecules of the cytokines. So this is the basic concept of cytokines, but we don't know the different types of cytokines. I mean to say different name of cytokines and their role, their source, their target cell. We don't know this. And with this content, I make a separate video. If you want to see that, hit on the I button at the right corner of the top screen. Okay, now I hope that the topic cytokines is clear to you. This is all about the cytokines. Thank you. Thank you all. Take care of yourself.